Welcome, everyone. Thank you for joining in today in this PHI webinar. I'm Lisa, Application Specialist at Phase Holographic Imaging, and it is my pleasure to introduce to you today our expert speaker, Kersti Alm, our Head of Biology. Hello, Kersti.
Hi there, nice to be here.
Today's webinar will be about cell movements and how to track and quantify your cells every step. If you have any questions during Kersti's talk, please ask them using the chat function. We will answer them at the Q&A at the end. Are you ready? Kersti? Ready, set, go.
Hi there, I'm happy to be here, and I'm really happy for Lisa to hosting this. What I'm going to talk about is how cells move. As you can see in this movie here, some moves a lot, other moves a little. Some mostly moves in circles, while other are long-distance runners. Cells move all the time. It's really necessary for the embryonic development and for the everyday function of all living organisms. Several methods are used to study cell movement. For example, scratch assays, transwell assays, Boyden chambers, time-lapse studies. They have all been tried since the 50s or something. Most of them are quite labor-intense, and in addition will affect the cells negatively when you use them. So what I would like to present today is an alternative way of studying cell movement. All our studies that I present here are based on digital holographic microscopy. I will talk about how to track the cells and the difference between cell motility and cell migration, and how to look at chemotaxis, how to use wound healing. And in addition, we offer that you get the cell morphology together with the cell movement. I will start with showing this nice image and discuss the difference between migration and motility. So if you see the starting point, A, the cells will go to B, but they usually don't go the straightest, shortest distance. They usually take a huge detour, like in the green line showing motility. What usually is measured in most methods is the actual migration, the shortest distance between the starting point and the end point. But what the cells actually have been up to is the motility measure. So I will talk a lot about migration results and motility results, and they can be quite different due to what you see in this picture. Okay, I think Lisa has a poll for you now.
Yes, so the question we want to ask you, just to, warm up a little, is what are your cells? Are they lazy couch potatoes? Or like energetic marathon runners? Do you have any idea what your cells are?
You know what I usually see, Lisa? I see that the cells in, say you have one cell culture, you will find all kinds of cells in that culture.
So like a multi-talent?
Kind of. See, even if the cells have the same genetic setup or makeup, what actually comes out is quite different.
Yeah, and we see here, like most of your guys' cells are multi-talents. So, wow, thank you all for answering. Yes, now everyone sees our screen again. Perfect.
And it's good to know that your cells behave mostly like they're multi-talent, but quite a few of you have really lazy couch potatoes. Almost a quarter of you have these lazy cells. That's very different depending on the cell's background and what is the purpose of that cell line, how they behave. In the picture that you're looking at now, or in the screen that you see right now, to the left there is a picture of the HoloMonitor. That's the holographic instrument that we use to make all the studies that I will show you, or we and our customers and all the users. The footprint of a HoloMonitor is about a sheet of paper, and you put it into the incubator. So the cells are quite unimpeded. They have the normal temperature, the normal pH, the normal growth environment, and all the studies are then performed just in what kind of cell culture vessel you usually use. 6-well plates, 96-well plates, whatever. To be able to analyze everything, we have the software, the app suite, that comes with a HoloMonitor. You need both of them to make the analysis. And the software is divided into 3 parts. We have the guided endpoint assays. There you just get the result like the cell size or the cell number or something, just one time point. For cell movement, that's not really useful. But we also have the guided kinetic assays, where you follow the cells over time, and it's all set up. So you just need to feed in cell name, treatment names, push the button, and the results will come up as graphs after the experiment has been run. There you can get a motility measure for how much your cells are moving. We also have the in-depth assays, where you get to dig through all the data and really set your teeth into everything. And there you can evaluate single cell movements and single cell morphological changes over time. You can get huge amounts of data. Okay, so how does this work, this fantastic instrument? I'm trying to just shortly explain how the holography works to make sense of what I'm showing you later. So to the left here, you can see that there is a diode laser. The laser beam will be split into 2 beams, where the sample beam will pass through the cell sample. It will go through the objective, and there it will meet the reference beam. The reference beam has gone undisturbed through the back of the instrument, so when those 2 beams meet again, they are not identical anymore. So they will create an interference pattern that we capture. That interference pattern is the raw data. That's what we use to calculate cell thickness, cell characteristics, everything. So what happens when the light passes through the cell sample, you can see to the right, the circle tries to show wave fronts. Those are the red lines. And when the wave fronts pass through the cells, you can see that there is an imprint, a shift in the wave fronts. It's called a phase shift. That's our rawest raw data, that phase shift. That's what we measure. If you have the phase shift and you have the wavelength of the light is 635 nanometers, and you have the refractive index for the cell and for the surrounding medium, then you can calculate the thickness in each pixel in your sample. So once you have that calculation, the software can create pictures based on that. So now in this screen, you see holographic images. To the left, you have the laser pattern. That's a hologram. These 2 blue images, the color shows the thickness, corresponds to the thickness. You can see that in the little bar, the color bar to the left. So you can see that the really dark blue in the bottom is the background. And then comes the white, which is about 2 micrometer thick. You can see the blue comes again, and it's about 5-6 micrometer. And then you have some kind of sky blue, which is the thickest part of the cell. And if you see to the right, you can get a 3D image based on this data. In this nice image, you can see there is a huge rounded up cell, which actually has all the different colors and which is the thickest cell in that picture. So all the images that you will see after this are these reconstructed images, and all the coloring put into them is artificial. So this is no labeling, no stains, and everything based on that shift in the light that happens when the light passes through the cells. Okay, so once you have these beautiful images, the next step is then for the software to go in and identify where is the cell and where is the background. So in the very most left image, there is a line straight over a few cells. And in that same image, you can see the profiles of the cells. So for the software, it's quite easy, based on these profiles, to find where is the cell and where is the background. If we try to do the same in a phase contrast image, you can see that the cell profiles look very different. It's much more difficult for software to sort out what is cell and what is background. That is the strength of having the thickness data in each pixel. Okay, so once we have found the cells, we can get a lot of data out. You can see to the left that for each single cell, there is a lot of morphological data that you can get out just because the software has found the cells. That means, like you see in the middle, that if you have an entire image frame full of cells, you get data from all these cells. And you get them presented in scatter plots, for example, you have about 30 different morphological parameters to look at. What we can see to the right also is that if we have cells captured several times after each other, like every 5 minutes, over time, you can see how the cells move because the software will be able to find and track the cells. So just based on simple image capturing, we get a lot of data, hard data out of the cells. In our software, we have 3 applications designed for studying motility. We have the Kinetic Motility Assay, which gives you the average cell population motility in real time. You get this, how fast the cells move, cell speed on average, and you get the accumulated cell motility. But that's only for population. You don't get the individual cell data out of that. For that, you need to go to spatial cell tracking. Using the software, the user will track each individual cell over time through all the image frames and get individual cell movement. How does it move motility-wise? That is, how far did it run? But also the migration and the speed, and if you have any chemotaxis, for example. But you can also connect motility to cell morphology. Then last, we also have the wound healing assay, where you get cell speed, cell migration, and you can get the gap closure on the wound healing. So you can combine the wound healing with the spatial cell tracking. So, oh no, there is another monster. Oh, poor guy, he got blasted properly. Okay, I think I will have to talk to Lisa. I think she put in all the monsters here, but that's great. Makes me happy anyway. Okay, so first I want to introduce to you the Kinetic Motility Assay, where you get the average cell population motility in real time. The output is both cell speed, so you get for every time point, you get the cell speed, but you also get the accumulated cell motility. So for every time point, how much the cells have moved is added. So here you can see the black will show you a control cell, while the orange and blue have been treated, so the cell populations move slower. And we have a publication on that. It's a group based mostly in Sweden, but also in the Czech Republic. They are looking at something called imprinted polymer particles. It's kind of plastic antibodies. And these particles affect cells. So different cell lines move differently to start with. You can see that in the diagram, where you have the accumulated distance versus time. And you can see that the MDA and B control cells, that's the blue line, they move much faster than the MCF7 control cells, always. And you can see that both cell lines are affected. SA-MIPs, those are the plastic antibodies. You can see through the dashed lines that both cell lines are affected, but totally different by the plastic antibodies. So this is a way to use the kinetic cell motility. Then there is the spatial cell tracking. I was talking about that every cell. Oh God, there is a cute little monster. And population cell motility and migration data can be given here, combined with morphology data. So the cell tracks to the left show how all the cells have moved over time. In the cell movement plot in the center, you get the tracks for each individual cell based on the starting point for that individual cell. So you can see here that most of the cells actually moved more or less in the same direction, but very differently straight there. You also get the cell morphology over time. Because you have tracked the cell, you can get the morphology out over time. So the right graph shows you how the optical volume of the cell shifts. And you see the straight dips. There are 2 very sharp dips. That's where the cell divides. That's kind of neat. I just wanted to show you that for this one single cell, for example, you can see the cell tracks to the left. That's how the cell moved during the time lapse. It's a 15-hour time lapse. And in the center, you can see how the movement is compared to where the starting point. And to the right, you see this optical cell thickness over time. And you can see that cell rounded up very sharply and then flattened out again. That's when the cell divided. So combining tracking, movement, and morphology is quite powerful. We have users that have used this, and I'm going to show you a couple of examples. I'm not quite sure about the pronunciation, but I'll try. Casciello and colleagues, they have been looking at tumor cells in vivo. No, tumor cells, sorry, not in vivo. But in vivo, tumor cells are prone to some kind of hypoxic state. Depends on where in the tumor they are and what kind of tumor it is. Most tumor cells are more or less hypoxic. So Casciello and colleagues, they're from Australia, they have been looking at a factor called CDH10, which is involved in hypoxia-mediated metastatic cascade, and it will down-regulate cell motility. And they have used another factor, G9A, which they studied, which will promote tumor cell motility, and that will then silence the first factor. So by balancing these different factors up, they can affect cell motility. And they used that. They used the MDA-MB231 cell line to see how different inhibitions and additions of these factors would affect cell movement. And you can see quite clearly that they have an effect on both the motility pattern, that's the upper colorful diagrams, and instead of putting those results in a plot that's used to make these target circles to see how far outside target cells moved, they also get the motility data over time for different treatments, and they could, as I said, determine that CDH10 is involved in the motility. We have also another wonderful publication, is from Korea, from Chirumarry and colleagues. They have been looking at anti-microbial peptides. They are quite interesting for cancer cell treatments, but they are not really efficient. So these Korean people have used synthetic molecules. They're similar but better, hopefully, to study cell migration. And they could see that treatment with their synthetic molecule would both change the locomotion pattern, that they call it here. The cell movement plots show you that the AGG2Es that they have been treated then with these peptides, they move a lot less than the control cells. That is also seen in the migrated distance to the right, where you can see that the cells that has this peptide move much less, even migration less than the control cells. Interestingly, there is a study where they have been used the cell tracking to see how individual cells change their morphology over time. This is an Australian publication from Croft and colleagues. I brought this along to show you that they can really separate out mitosis, which is the upper left diagram, from cell death, which is the atoposid and the staurosporine diagrams. In the DKL-SO2, you can see that first they have mitosis, normal cell mitosis, and then they have cell death. So using the single cell tracking and morphology, you can get detailed information about your cells. As I mentioned before, the movement can also be used to study chemotaxis. So Szatanek et al. From Poland have used the HoloMonitor to study chemotaxis. Cancer cells will expel small microvesicles. Those microvesicles are usually intended to affect the immune system cells, but they also affect themselves. In this study, gastric cancer cells expelled these microvesicles, which then affected the cells to start moving in a chemotactic way in a certain direction. This is usually not considered good for metastasis. Another way to study motility is looking at motility assays. Wound healing assays, where you can study gap closure and where you can track cells at the same time. So here I picked out 3 pictures, the beginning, the middle, and the end of a time-lapse movie, showing how the gap is closed. And to the right is the corresponding diagram showing the gap closure. But because it's so pretty to look at, I just have to show you this movie. This is how the gap is actually closing. And you can see that the cells are moving very differently. Some move almost like a cell front, while others more moved like individuals and just got ahead of all the others. This assay has been used to study the effect of, actually, this is quite interesting, of HUVEC cells, they're umbilical vein cells, embryonic cells, and how they are affected by mesenchymal stem cells. So Zhou et al. In China have used the wound healing in the HoloMonitor to study this. And you can see in the pictures that to the left, upper left, there is, to the left behind the red line are the mesenchymal stem cells, and to the right behind the green line are the HUVEC cells. And you can see that if you follow that upper row towards the right, you can see that HUVEC cells are slowly moving towards the stem cells. In the bottom row of pictures, they have inhibited a factor called KFL2, which is usually used for which is implicated in pluripotency and proliferation and other cell processes. So they have inhibited that factor or silenced that factor. And you can see that now the HUVEC cells are more eager to move towards the stem cells, but also that the stem cells, the mesenchymal stem cells, are slowly moving towards the HUVEC cells. So both that can be clearly seen in the wound healing assay. You can also see in the diagrams below that the migration speed is different with and without the factor. And you can see that the distance is closing at different speeds with and without the factor. In Israel, Varan and colleagues have also used the wound healing assay, but they combined it with a tracking assay. In the upper frames, you can see the wound healing and the gap closing after 0 and 24 hours. And there is something called the long non-coding RNA, which is an RNA that affects cell function even though it doesn't produce any proteins. In this study, they looked at the TP73AS1 and how it would affect tumorigenicity in medulloblastoma cells. And you can see that below the images showing the wound healing gap closure, you have the movement plots, and you can see that the left plot has a 300 micrometer axis, while the two right plots have shorter axes, 142 in the center and 171 to the right, showing that when the TP73, the long non-coding RNA, has been silenced, the cells move less, quite less. And if you look in the diagram row below, you see the mean migration is affected as well by this silencing. The black is the wild type, and you can see in the center diagram there that the motility is not affected. So the cells move just as much, but they don't move as far away. So using combined tracking and wound healing, you can pick out interesting data that would otherwise get lost in the population. One of our users, Zhang, together with Judson in the US, have looked at the motility as measured by the HoloMonitor compared to transwell motility measurements. They have used both the motility, the kinetic motility, and the wound healing assay. In the upper right bar diagram, DHC motility is the kinetic motility, while scratch is the wound healing assay. And they compared that to transwell assays, one for migration and one for invasion. And they used two cell lines, one which is non-metastatic and one which is highly metastatic. And both cell lines kept their comparative movement, or showed the same comparative movement, independent of what assay was used. But in addition, with the HoloMonitor, Zhang and Judson found that there were a few highly they call them hypermotile cells. You can see that in the diagram below, where each individual cell motility is shown, that there are, in the highly metastatic cell line 1205LU, there are hypermotile cells moving much faster than the rest of the cells. So in short, oh, hi Monster. Oh, you got really happy to see me, right? So cell movement in short. Many cells move all the time. Cell movement can be random or directed, and it can be measured by the HoloMonitor. You can distinguish migration and motility, and you can get the speed, and you can get the direction, and you can have the morphology all in the same experiment. Thank you so much for listening, and have a nice day.
Thank you, Kersti, for your talk. I hope everyone had a great time listening. Now it's time actually for our Q&A. As I've seen, we're actually quite at the end of the webinar already. We just picked like one of your questions to answer now, and we will answer every other question by email. So please just use the chat function to ask us your question. We will answer it. But for now, I'm just going to pick one. So we're going to, so we have Martin here from Vienna. And Kersti, he's asking, can I track any type of cell?
Well, the HoloMonitor is made to work with adherent monolayer cells, but anything from cancer cell lines to primary stem cells work. If you have questions regarding a specific cell type, please contact us. Also, you don't need to use the chat function here. You can email us later or contact us on our webpage.
Yeah, anytime, contact us. If you have more questions, you're always welcome to contact us. Send us an email to info@fiap.se. And you can also find a lot of information on our HoloMonitor and applications online. So you can always stay up to date to our latest news, activities, and events, online events. With that, thank you for joining today. See you next time. Hej då!
Hej då!